Hi everyone, welcome back to another episode of NeuroSciQ. This week you're in for a treat. I've been asked to give a talk on how artificial intelligence can accelerate our ability to biobank and do translational research using clinical samples. And I pre-recorded it so that all of you guys can watch it while I'm delivering the presentation on October 4th. So get some popcorn, sit back and enjoy the presentation. Hopefully you learned something new. So hi everyone, my name is Diamandis. I'm a neuropathologist at the University Health Network, and it gives me great pleasure to be here and talk about how deep learning can accelerate and improve our practice of biobanking and translational research discovery. So I have no conflicts of interest to declare. And really to put this presentation in context, I'm going to tell you what our translational research workflows look like and where biobanking fits in. We usually start off with a bright idea or not so bright idea where we have a hypothesis about what question we're trying to answer. And then based on this, of course, we will contact our local biobank and try to get the appropriate specimens to answer this question. Once you get your specimens, you're going to do a pathology review, which entails setting up an appointment with a pathologist and asking them to identify what you're looking to study under a microscope. And then, of course, as most translational research questions are now answered, you use some sort of omics technology to study the whole genome or whole proteome in search of biomarkers to find predictors of your specific subgroup of interest. And for the most part, I think most people will agree that steps 2 and 4 have been made particularly easy in recent times due to massive improvements in how biobanking is done. It's relatively new activity, and there's been massive improvements in the number of specimens and our ability to store high quality specimens, and especially at our institute, the staff there are exceptionally good at finding the specimens you want in a relatively easy manner. And of course, omics technologies are getting cheaper and faster, so those are really what I consider the easy steps of any translational research question. The hard part, I think, especially in 2019, 2020, is coming up with an original research question that hasn't been asked yet. That's actually quite difficult. And of course, sitting down with a pathologist and objectively trying to annotate the tissue that you're going to analyze. So the outline of this talk is really to show how we can automate some of those challenging components of translational research using AI. And there's 2 key components that I want to talk to you today. One is automating the pathology review. Can we automate specimen annotation and review using what I call supervised learning or classification tools? And then the perhaps the harder part that is still a work in progress, can we annotate some of the anomalous pattern detections and proposals that are really the fuel that drive novel research ideas? And to do this, we really need to shift our AI strategies and use more unsupervised and clustering techniques. So let's look at how we're going to fix the pathology review, or at least use AI to accelerate it. So this is what I think pathology review looks like today for the most part. there's a number of different tasks that we're asked to do, or at least I'm asked to do as a pathologist when I'm involved in a research study in a collaborative form. I'm usually asked to confirm a diagnosis. I'm usually asked to estimate cellularity and perhaps circle areas of the slide that are highly cellular and useful for molecular profiling. And in some specific cases where the question revolves around tumor heterogeneity, of course, there could be instances where I need to circle both the epithelial and stromal component of a tumor. And these, of course, are done mostly with a magic marker, as shown here, as I'm sure most of you know, which is quite outdated in today's digital society. So how have we tried to modernize this process? Well, for a long time, the goal was to use a branch of computer science known as computer vision to try to automate some of these processes. And this really involves taking a slide as shown here and working with a pathologist to identify key diagnostic structures that would be of diagnostic importance. And then here you have your feature engineer or your computer scientist try to recapitulate some of those microscopic features into lines of code that could be reliably extracted from images in the future. These classifiers are then used to classify these images and give a diagnosis, and that's really what a computer vision workflow has looked like for quite some time. So how do we know how good these computer vision workflows are? Well, you can test them, and for maybe the last 7-8 years, there's been a competition known as ImageNet, which is basically a collection of 1.2 million images in which computer scientists meet and compete each year to try to see which algorithms are most effective at pattern recognition. And the 1.2 million images usually spanned 1,000 different classes, as you show, that are commonly seen in everyday life. So cats, dogs, butterflies, cars, planes, and really the goal here is to see which algorithm makes the fewest errors in identifying the objects. Now you can see why this could be quite difficult. Here's a picture of a cat and it's hiding behind what appears to be a sofa. So if your computer engineer relied on whiskers as a feature to look for at defining a cat, you can see that that would be quite erroneous. And of course, this butterfly here has this pattern that could resemble eyes, and these could resemble ears. So you can see how this manual process of feature engineering could result in errors. And in fact, when you look at the error rates of the kind of initial entries in 2010 when this competition started, there was high error rates of about 25% in the first 2 years. And you can see just to kind of orient yourself, a human doing this competition would make an error rate of about 3%. So these traditional tools, at least at the beginning, were very far off from what a human can do in this same task. So around that time, there was these transformational improvements in another form of computer vision technology known as convolutional neural networks. And this is just a picture showing how the workflow for these deep learning tools looks like. And the idea here is that instead of trying to manually engineer these discriminatory features, you can instead take advantage of large image data sets that are labeled. So you can see here in this pathology example, you would have these snapshots of different tumors that are labeled with different values. And by running those through this algorithm, the algorithm could analyze groups of images from a specific class, pixel by pixel, and try to find specific pixel patterns that are common between the images of that class. Now, the beauty of this model is that in the early layers of this convolutional neural network, it will first try to find elementary features like lines and particular colors and edges. And as these are passed through this multi-layer network, these will be aggregated together to form more complex features that are perhaps unique to a particular class. And because of this kind of hierarchical arrangement, as features are passed through and through, the number of combinations of features that you can generate get to the order of millions, which is far superior than what you can do by manually creating each and every individual feature. So once you've trained your neural network and developed these features in what's called a data-driven manner, you can then present to it new images, which it will again feed through these multiple layers and again look for, at the basic level, lines and dots and circles, and it will start aggregating those again to create complex objects that are of diagnostic value. And you can see here we have a test image of a schwannoma that's run through, and again it will look for the specific patterns that are learned from the training data, and you can see in fact this was classified correctly. And here's just kind of a cartoon of what this may look like at the individual layer where things are aggregated together to form more complex objects. So this, of course, was pioneered by computer scientist Geoffrey Hinton at the University of Toronto at the time. And when he entered the competition in 2012, he introduced this deep learning model. Again, it's denoted here green to just to kind of signify that these are different types of computer vision algorithms. And you can see that, in fact, there was a quantum improvement in the error rate that was the winning error rate for that year. And this really caused a transformational shift in how computer vision was done, and now almost exclusively using these deep neural networks. So subsequent years, again denoted here by green, were improvements of his original architecture, and you can see that the error rate continued to creep down lower and lower. And in 2016, these neural networks, at least in this competition, in this ImageNet competition, now match or even exceed the capabilities of humans. So of course this didn't go unnoticed. There's a number of areas of our life where image recognition is so very important. Driving cars, and of course in medical image analysis. So there's been a proliferation of deep learning applications, especially in medicine, where deep learning provides an objective way to carry out many of our kind of pattern recognition tasks in medicine. And here's one of the kind of early transformational papers where neural networks were trained to identify skin lesions at a level that matched or even exceeded dermatologists. And here a more recent paper shows that you can even look at the patterns of the nuclei to predict mutational statuses in non-small cell lung cancer. So what does this look like on a practical level? So here are some of our work, just to show how this process works and just to show how robust and reproducible this really is. So here's an H&E slide of a resection of a glioma. We run these through a trained neural network, and you can see here we get these CAM or class activation maps that show you where in these individual fragments the computer detected tumor, which is shown here in brown. So obviously there's a heterogeneous distribution of tumor within this specimen. Now, how do we know that the computer is actually picking it up where the tumor is? Well, you can use this IDH stain that stains specifically for tumor cells, and you can see that where the brown is on this immunostain corresponds very, very nicely with the class activation map developed by the computer, showing really just how sensitive and specific these computers are at detecting tumors. And here again is another representation of how a CNN can find tumor cells exactly where tumor is expected to be. Here we see an H&E of an infiltrating glioma, and you can see that even when you zoom in on some of these areas, you get very, very high resolution of this infiltrating pattern. The computer is able to detect tumor cells even within normal brain tissue. So quite remarkable technology for pathology. And what you can do now is that once they're trained, these become relatively routine to generate. So you can generate these gigapixel sized images and convert them into these nice, beautiful cartoons that tell you exactly where and what type of tissue is found on the slide, and in a somewhat quantitative manner. So you can see from our workflow, from our translational research workflow, this really is a refinement of how we look and analyze and annotate slides. So how are we using this in some of our work? Well, we're interested in intra-tumoural heterogeneity, and one of the things we're trying to do is see if we could use AI to find subtle changes in the morphologic patterns of tumors and use those as a stepping stone to look at intra-tumoural heterogeneity. This is one of our earlier class activation maps, which has much lower resolutions than the ones I showed before, but it's suffice to say that what we see here is that there's these patches of this tumor that appear to be lower grade and higher grade, which now allows us to analyze them to see if there's any changes either in their genomes or their proteomes, which is the technology of our choice. So these now become very objective and reproducible ways to annotate tissue and look at morphologic or feature differences for intratumoral heterogeneity and molecular analysis. Now the beauty is that you can do this on a large scale. So I know when I deal with tumor banks, I'm often given these surgical pathology reports, and of course as a pathologist these are quite useful. But imagine we can now create more user-friendly reports for non-pathologists where we have a diagnosis, we confirm the diagnosis here. These are some of the report structures that we're trying to optimize for things like biobanking, where we provide a confirmation of the diagnosis, hopefully, and then we provide these heat maps here that tell you where the computer used to analyze the tissue, and these are all brown because this is all tumor. You also get a nice picture to accompany your report, so you know what the tumor looks like at high magnification. This is one of our more optimized reports that has much more information. Again, it has a diagnosis on the first page. It tells you what molecular analyses should have been done at a very minimum to help guide you in your tissue selection. So you want to pick cases that have these stains done, as they'll be much more valuable when it comes time to publish, that you've extensively vetted the molecular patterns that are known to exist in these tumors. Again, you have this second page that shows you where the tumor is, the approximate surface area of the lesion, so you can kind of plan your molecular analyses much better if you know how much tissue you have, and of course there's quality control metrics to try to standardize this over multiple days and multiple different tumor types. Now, obviously, we're interested in working with biobanks to completely create a catalog of all their cases. This would be a a nice long-term project that we're excited to hopefully find a partner to help fund and collaborate us with on this effort. But in the meantime, we're also trying to develop a website where someone with a few individual cases can take advantage of some of the neural networks that we have available to annotate their tissue prior to their omics analysis. So this is kind of what our website looks like. I'll show you one of our kind of older demos that just kind of highlights how easy this process is. Here's our website again. You click the particular app that's relevant to your tissue type. You take a slide that's available on your computer. You drag and drop it onto our website. This gets uploaded. You can also upload clinical information that you want in those reports. You add your email where you want your reports sent to, and then by clicking generate, this will send your image to our kind of server and carry out this classification in the cloud. Once it's done, it'll send the report to your email, and then you can print or save it as a PDF, and again have this information readily available to you. So if a pathologist is not available, if they're too busy, you now have an alternative way to do the annotation and diagnostic step in a relatively automated way, and we're really excited about that to launch that once we have the appropriate collaborators. So hopefully I've convinced you of how AI is going to be a transformational tool for the automating the pathology review. A much more difficult thing is, but a much more exciting thing for us is trying to use AI to come up with some unique and interesting proposals of projects that we could actually use our omics and rich biobanks to answer. So let's talk about that and how can we achieve that. And really there needs to be a fundamental shift in how we do our AI. Most of the AI that we've talked about today is what's called supervised AI, where we have a specific output in mind. And that's analogous to this is a nice example to show what supervised learning looks like. You have a particular input, so you have these Lego pieces. They're colored, of course, so we can call them labeled. And the task that you give your small child is to try to align the colors of the Lego pieces with the particular colored paper bags to meet that are congruent. And of course, the beauty here is that you can see if someone can replicate this task, and you can grade them in terms of how many red Lego pieces are in the red bag compared to the other bags. So this is what a highly supervised learning or classifier looks like. And really the goal here is to classify things and replicate a defined task. And as we saw, computers are very, very good at this, and we've developed workflows that are exceptionally good at this process. What unsupervised learning attempts to do is to not provide explicit instructions. Instead, you'd let your baby play until it realizes that there are particular patterns in these Lego pieces, and instead of trying to group Lego pieces based on their on these paper bags, it will eventually group them into these empty bins based on their color. The advantage, of course, here is that, as you can see, there's some white and black Lego pieces, and these don't really make sense to go into any of these paper bags. So the beauty of unsupervised learning is that when you have these anomalous exceptions to the rule or cases, you don't need to erroneously classify them into these kind of rigid outputs. So it allows you flexibility to find outlier cases and actually detect them with some confidence and actually do something with them. The other beauty of unsupervised learning is, of course, that there's really no particular output that's required. So one child may want to group things based on color, but another equally creative child may want to group things based on the size of the Lego piece. And that's really the beauty of unsupervised learning, is that there's a lot of flexibility for creativity and for eureka moments that I think could really drive innovations in research. Just to show you how important this type of learning is, here is a pioneer of AI, Young LeCun, who basically phrases it like this. Most of human and animal learning is in fact unsupervised learning. If intelligence was a cake, unsupervised learning would be the cake. Supervised learning would be the icing on the cake, and reinforcement learning would be the cherry on the cake. So you can immediately see that if you're interested in replicating human thought and automating at least manual aspects of its creative aspect, you really need to focus on unsupervised learning. So what does unsupervised learning look like? The outputs are fundamentally different. So these are what supervised outputs look like. So supervised tasks would be, for example, of continuous variables would be to try to model the price of a house depending on the size of the house. And obviously, as we know, most houses will increase in value as the size of the house increases. More discrete classification kind of task would involve our email clients. Can we predict if an email is going to be spam or not spam based on what domain it's coming from? Is it coming from an Outlook.com or is it coming from a unknown domain? And is there specific keywords that are commonly seen in spam emails, such as win $1 million, right? So this would be a discrete classification task to either send an email to your inbox or send it to your junk. Unsupervised outputs are much conceptually different. Instead of having a conclusion, they instead arrange data into groups, and this can be done either as in a continuous way using kind of dimensionality reduction plots like t-SNE as shown here, or principal component analysis. And the goal here is that most of these plots have some sort of spatial relationship. So the closer two groups are, suggests that there's a closer relationship between these two compared to clusters that are further away. If you wanted to discretize your classification or your grouping, you can use hierarchical clustering, where you get a nice ontological grouping of how each sample is related to one another, and you can create these larger groups and clusters that group your seemingly unrelated data into these groups that have similar patterns of these different features. So these are the tools that we use for unsupervised learning. So let's look at some of those based on some of the work that we're doing. This is again an example of a supervised classification task. You train a neural network, then you pass an image through, and you get a very discrete outcome where it classifies it based on what you've learned, of what you've taught it. In unsupervised learning, instead of letting the image go all the way through, we're more interested into how this image registered at these different nodes within the neural network. And because, as we said, the deeper you go into the neural network, the higher the level features are. Going near the end of the neural network, or in the final last layer before this classification occurs, you get the kind of most complex features that the computer learned about your data. And again, you can use these tools to see the relationship to other components of your data. So you can see that this t-SNE is meant to reproduce this this binning approach, where you can see these individual bins, and of course there's space in between here where other bins can also be inserted if need be. So let's look at what that looks like. So here is a test case of an anaplastic astrocytoma. We want to classify it and understand its relationships to other lesions. So what we can do is we can take these randomly selected areas and plot them on this t-SNE plot. These diamonds represent these test images, and what you find is that in fact these test images overlay quite nicely onto this blue cloud or blue cluster, which is your glioma cluster. And so you're able to, with confidence, say that this image is likely a glioma based on the fact that the images overlap with the test images of the training set. These are blue Lego pieces going into a blue bin. The beauty of this t-SNE plot is that when you encounter a lesion that perhaps was not in your training data, rather than it forcing it into one of these bins, as you see here, it actually put it in this no man's land. Here's a zoom in, suggesting that this is again an anomalous lesion, and instead of having to force it into one of these groups, it instead will make a new class and say, this is perhaps something of interest. This is perhaps something that you should study more closely because it's unlike the ones that you've studied before. And in fact, this was a gliosarcoma. This is a rare subtype of a glioblastoma that has a sarcomatoid component, as shown here by the reticulin. And so really showing you how you can use this to your advantage to scroll through many, many cases in an automated way and find interesting cases that are perhaps more interesting to invest your resources in than things that have already been studied. So moving on, what about larger scale studies? Is there a way to group large collections of patient data and try to find underlying patterns and group them in a kind of coherent way? So we wanted to develop a clustering approach that you can use neural networks to cluster your different cases and see if we can do this in a way that resembled kind of intelligent human thought. And to do this, the first problem we picked was to try to see if we can recapitulate the WHO classification guide of central nervous system tumors. This is the World Health Organization's the kind of manual to how different brain tumors should be grouped or at least conceptualized. So you have different types of meningiomas that are grouped in this meningioma chapter. You have different types of gliomas that are grouped in the glioma chapter. And of course, these are based on the consensus of international experts that have met to discuss and vet similarities in the biology, the molecular patterns of these tumors and the clinical outcomes, and kind of organize it in a very coherent and intelligent way. Pathology exists because many of these biological behaviors often correlate with microscopic features, whether it be the way the nuclei are arranged, kind of spindled patterns, whorls in meningiomas, rosettes in specific types of glial neuronal tumors. And so we have nice histological correlates that help humans organize these different types of tumors. Now, of course, there's some features that are common between malignant features like high nuclear cytoplasmic ratio that's found in a lot of different malignant tumors. But overall, there usually is some specific features that are unique to specific classes. So what we did is we generated close to 1 million images, about 850,000 images of approximately 72 to 78 classes, I believe the last count we had was. And then we asked the computer to use clustering based on these kind of 512 features found in the deep areas of the neural network to group these different tumors in a kind of ontological or family tree fashion. And what we found was that, in fact, computers are able to find these subtle histological features that humans use to group these tumors together. And these are just some examples. Again, we had, this is a cluster of of 8 tumors that the computer thought looked very similar, and you can see 7 of the 8 tumors here are in fact meningiomas. We didn't explicitly tell the computer that there was multiple types of meningiomas. It kind of sorted out on its own that these tumors should be grouped together based on similarities in their histology. So we think that really a lot of this kind of intellectual work can be automated by computers. Interestingly, here we have one of our kind of misgroupings, you can say, where you have a meningioma clustering with this other tumor called a hemangioblastoma. While this can be considered wrong, again, it's interesting to note that both these tumors are vascular tumors. So perhaps the vascular component of these tumors dominated the clustering and grouped those together. So even the errors that we see are not, in fact, kind of unintelligible. There is some kind of reason why the computer thought these tumors should be grouped together. And here's just another example of another cluster where the computer intelligently grouped together these different types of gliomas, diffuse gliomas, which of course is also equally as impressive. Interestingly, the lower grade gliomas form their own kind of family of tumors, while the higher grade gliomas grouped in this second group, admittedly with lymphoma, again because of those similar features of malignant tumors, such as the high nuclear to cytoplasmic ratio. So this is a very good way to automate some of these very challenging tasks that require consensus from many pathologists. Now, the nice thing about grouping these tumors together is that we can find these specific nodes in the neural network that are common. So you can see here, this particular feature or node in the neural network lights up whenever a metastasis is noted. It's a metastasis group. So we can use, we can actually use that node to kind of colorize on this image where of the map it's most activated on. And you can see here, we can start to learn how a computer is organizing these specific cases. And as we see here, it's in fact doing it with the same features, whether it be the epithelial component or the luminal component, as many humans do. And the importance of this is that we can now build confidence that by automating some of these processes, we're in fact recapitulating how humans learn and classify these lesions. And here's another feature which was more common across all tumors. This is the feature 215, and you can see here this labels more the fibrotic aspect of this slide. So it wouldn't be fair to present to you for such a long period and not talk to you about a tumor related to the GU system. So we thought we'd present some of our unpublished work on renal cell carcinoma. And here we're trying to really create generalizable tools that can be applied to other types of tumors without the need to recreate millions of images and train these neural networks, because this process can in fact be quite cumbersome. So we wanted to see if we can carry out some intelligent tasks outside of our field using these neural networks that have learned, that have been educated on a diverse array of features across brain tumors. So we took 60 renal cell carcinomas, 30 were papillary, 30 were clear cell, and we asked, can a computer that's been trained on brain tumors differentiate these two different types of tumors in a way a GU pathologist would? And in fact, as you can see here, for the most part, it was able to organize them in a somewhat intelligible way. There are, of course, some misclassifications, and we're working towards trying to understand why these errors were made. Was it that this is in fact the papillary clear cell variant, of this variant that could have gone both morphologically, or is this in fact an error? So, but overall, you can see that it's able to subtype these different types of tumors with really no knowledge of being trained in GU pathology. So we thought that was quite an impressive feat. To take it one step further, we said, okay, well, if it can differentiate the biology of different types of renal cell cancer, can it differentiate distinct biology within a single type of renal cell carcinoma? So we took 399 cases of clear cell renal cell carcinoma from the Cancer Genome Atlas and asked our kind of brain tumor educated classifier to group them based on the morphologic features that were shared across different types of tumors. This is all preliminary, but we found was if you take the early bifurcations of this dendrogram, and you use the three major groups that were identified based on these kind of 512 high-level morphological features, they do in fact have survival differences. The patients, you can see that there is a small group, albeit that have a relatively good prognosis. We have a kind of aggressive subtype of clear cell renal cell carcinoma, and then you have kind of a moderate group, a group with a moderate outlook. So we think that this is a way that you can in fact automate class selection or tumor selection to find interesting biological patterns that may be in fact interesting to analyze using omics technologies. So hopefully what I've convinced you today is that we've taken this translational research workflow and have tried to address some of the kind of cumbersome or human components of the translational research workflow, and by using both supervised and unsupervised techniques, we're able to automate and simplify this process to create a really fully automated system. So I'll stop there. I want to acknowledge the whole lab, especially Kevin, who is our chief programmer, and Adil, who's working on some of the work in the GU that we presented today. Really everyone. Ugi is kind of the co-director of the lab here, but everyone for their inputs. Of course, we want to thank our funding agencies for believing in us early on and allowing us to grow and ask these interesting questions. And if you're interested, you can rewatch this lecture on YouTube, available immediately. So with that, I will stop and thank you for your attention and hope to see you next time. So thanks for watching. If you made it this far, thanks for watching. We're always looking to grow, so please hit the like button, leave a comment down below, tell us what you think are the kind of killer applications of artificial intelligence, and please tell your friends to check out our channel. See you at the next one.